uit.
Ja,
Mensen, gewoon met
support.
veel mogelijk mensen. Dank je.
Ja, wel jongens een beetje snel, want vorige keer gingen we echt snel.
Is goed. 1, 2, 3 en staan. En als ze het fout hebben, gaan ze zitten.
Als er 2 opties zijn, is het staan zitten. Als er 3 opties zijn, is het gewoon hand in de lucht wat het is.
Ja.
1, 2, 3.
Ja. De olifantvraag, de vulkaanvraag en de bestekvraag. Maar dat vond ik een beetje stom.
Ja, fair enough.
Ik weet niet zeker of ze weten.
Dus
Dit is een nacht.
deze mensen zijn koude intelligent. Dus
Ja, waarschijnlijk wordt het easy peasy lemon squeezy. Hebben we nu ook een cadeautje?
nee, ze hebben mij
Een marshmallow.
liefde.
Ja, per.
Hey, deze, ja, ik wou zeggen, wat is vaak gebeurt hier? Allemaal Nederlanders.
Echt?
Oh,
Test. Dat was heel hard. Hallo? Hallo?
everybody can come inside, please, now. Thank you.
Attention, attention. Please mind your step. This is the last call for the flight no 15AB. The boarding will close in 15 minutes.
Sorry, Bert.
Prima. Prima, is ook niet zo goed.
Huh? Siggy? Wat is dat?
Siri.
Oh! Wacht, hoe gaat hij ook alweer? Wacht, hoe gaat hij ook weer? Hi,
Wat
Siri.
wil je zeggen?
Ik weet niet wat je wil.
Ja. Jij moet zeggen hi Siri en dan moet ik zeggen hello Richelle.
Nee.
What can I help you with?
Nee.
Today.
Today.
Nee, stop.
Huh?
Smallen.
Hoe is Smalland? Oh ja.
Nina en Joost willen graag opgehaald worden uit Smalland. Nina en Joost willen graag opgehaald worden uit Smalland. Joost, kom gewoon even in mijn boven.
Attentie lieve klanten, de Albert Heijn zal binnen 15 minuten sluiten. We verzoeken jullie zo vriendelijk mogelijk om de winkel te verlaten en jullie boodschappen af te rekenen.
Eerst de boodschap afrekenen.
Wat? Hoe gaat die dan?
Ik ben nog nooit bij sluitingstijd
Ik ben nog
geweest.
nooit... Oh!
10 minuten.
Attention please, this building will be closed in...
Oké, dit is klaar.
Die error, die mevrouw van de MF heeft een gekke error als ze in het Engels praat.
Is everybody here now? Okay. Everybody's here? Okay. Okay. All right, ladies and gentlemen. Good afternoon. My name is Richelle.
Wacht, geef ze even tijd.
Dit is iedereen.
Ja, maar niemand let op, dat bedoel ik. Kijk, wacht, je moet het zo doen, kijk. Hello everyone, welcome in this room again. We're kindly waiting for you to have attention for us.
Ladies in the back, we're trying to get it started.
With the quiz.
Begin maar gewoon, begin maar gewoon.
All right. We like to tell you something about the Netherlands today, and we would really like for all of you to stand up because we're going to do this in a more interactive way. We have prepared a quiz for you, so I hope you prepared your knowledge about the Netherlands. So stand up, guys. Yes, you guys. Very good, thank you. All right, so who are we? We are some students from the Netherlands, and we study at the VU, the Vrije Universiteit. We are third-year medical students, and we want to tell you something about ourselves. So let's start with the first question. What city are we from? Show us your hand. Give me the answer. Amsterdam. Very good. So there you go. It's very easy. So if you have the question wrong, please sit down so we can see which one of you are left over at the end. And they get a big hug from us. All right, the next question. What is the famous football club of Amsterdam? And I want to see A1, B2, C3. So put up your hands, show us. Show us your hands, guys.
High and clear, please.
High and clear. Yes. I see a lot of twos. What is your answer? 3?
C?
All right, so it's B, Ajax. So sit down. Sit down as well, miss. Thank you. All right. Dan, ga hem vragen.
Ga er maar voor.
Iedereen praten?
Vraag maar.
Alright, the next question is, what is the national flower of our country? So, does anyone know the answer?
Put Put up your hand.
Put up
who
your
knows?
hand.
In the back. Tulip.
Correct.
Yes, correct.
Good job.
But there is a little there's a little side note to this answer, because honestly, it's not from the Netherlands, it's from Turkey. So actually the answer is wrong.
But we capitalized it. Like most things. All right, so the next question. What are the colors of the flag of the Netherlands? Yes. Which order? Well, perfect. Yes, it is.
Op welke ding het lijkt, op welke land?
Oh, and which
Question.
bonus question? To which other flag in Europe does this flag look very similar? Which country?
Yeah, okay.
Turkey? Germany? Germany? No, that's yellow, black, and red.
France.
France, exactly. You just have to turn it 90 degrees and then it's France. So the next question, what kind of currency do we use?
Yeah.
Euro, yes. So 16,900 rupias is 1 euro, so we have to do a lot of math these upcoming few weeks. But we're becoming very good at it. All right, volgende vraag.
All right, people. So I know here in Indonesia we eat a lot of rice and a lot of meat and a lot of spicy food as well. But in the Netherlands we do it a little bit different. So what do we eat for breakfast? Is it A or 1, bread, 2, nasi, and 3, pancakes? I want it high and clear because I can't see enough. Okay, I see lots of A. Ik bedoel A. And that's obviously correct.
It's bread. So everybody has 3, sit down. Yes, thank you.
I like it with peanut butter, it's the best.
Yes. Or yes, chocolate. Nutella. Yeah. All right, so the next question. As you've probably noticed, we are very tall. So what is the average height of a Dutch man? A is 1 meter 68, B 1 meter 75, C 184, or D 1 meter and 90 centimeters. So give us your answers. I see 2, 3.
Ooh, there's division.
2, 3. Woo. A lot of people are gonna sit down after this. So the average is 184, so C. Yes, it's very tall, like I said. So here you can see the big person on the left is actually the average height of the Dutch male, and the rest is the rest. Indonesia is also there. Right, so the next question, what is the temperature in the Netherlands today? So minus 5 degrees Celsius, 10, 17, or 30?
Oh, I say
2.
1, 2, and 3.
Yes. All right. Okay. So it is actually 10 degrees Celsius. It's very cold, and a lot of rain and storm. So this
We
is
are
way
glad
better.
to be here.
Yes. This is us every day. Rain.
On the bike. All right. So this is a little knowledge question. In Dutch we have some similar words to Bahasa, so we were wondering if you know which word. So what word is the same in Bahasa and in the Netherlands or in Dutch? Is it enak, handuk, sepatu, or meja?
Yes. Handuk.
1,
Very
2,
good.
3, or 4.
Okay, yes.
Amazing. So thank you. You are the winners of this quiz. And then you can sit down, guys. Thank you for your participation. And we'd like to tell you a little bit about our school day, because we were very curious about your school day, but we'd like to tell about ours first. So we start our day with some bread, as we told you, very delicious. And then we go to our university by public transport or bike. There are average of 2 bikes per person in the Netherlands. So it's huge. There are more bikes than people. And after this crazy drive through the city and almost being run over by multiple cars, we arrive at the VU. So this is our campus. The red arrow points to our main building. It's called the Medische Faculteit. There we have all our classes, and we also have one huge lecture hall in below this hospital. So the patients are rolled out of the wards, put down in the basement with us in the lecture hall so we can hear their stories. It's very nice. And here you can see on the left a picture of that lecture hall where patients are being rolled into. And on the right you see our medical faculty where we can study, have lunch, have some fun, study a lot. So after the morning lectures we usually have lunch, also bread, maybe a bit of fruit, no nasi, sadly. And then after a hard day of school, we usually all have jobs, so we work in restaurants or supermarkets. It differs, but everyone has to pay their rent, so we all work pretty much a few days per week. And we also like to sport, so at the gym or hockey, football, anything like group sports are very popular as well. And after that, on the left, you see stampot boerkool. It's a very famous dish in the Netherlands. It's very delicious. I can't wait to try it again when I'm back home because it's like mashed potato with greens and a sausage. And at 8 o'clock we also always have tea and coffee time. In our tea and coffee we don't usually add sugar, so it was very fun to taste your tea here. It's very sugary. And at the right you can see our houses. They're a bit different from you guys as well. So, terima kasih. This is what we want to tell you about the Netherlands, and thank you for having us here.
Yes, thank you so much. So, can you mention your name, guys?
Oh,
My name?
Yes.
I'm Marwa.
Marwa?
Daniel.
Daniel?
Richelle.
Marwa, Daniel, Richelle. Thank you so much for entertaining us. So, you may back to your seats and hopefully just make a Netherlands one of your target wish list for your study or your just for the tourism place. Okay, so everyone, welcome back to Alright, so everyone, welcome back to the summer course 2023, and now we are going to take part of the first panel for the summer course that will discuss about the pharmacogenomics. In this panel, we have around 4 speakers from national and also overseas speakers. But before we join the first speaker of panel 1, we will have one left keynote speaker that already joined with us using Zoom from Swiss. And I would like to invite the moderator for these sessions. I would like to invite Ibu Firdhani Satia Primasari S.Farm Apothec M.Clin.Pharm from Department of Pharmacology and Clinical Pharmacy, Faculty of Pharmacy, Universitas Gadjah Mada. For the next around 3 hours, I will give the stage to Bu Tia.
Okay, yes, good afternoon everybody. So here for the next 3 hours, I will conduct this discussion about the pharmacogenomic as said Mas Yopi before. Before we start the session, there will be the second keynote speaker from the Dean of the Faculty of Medicine. He is Professor Yodi Mahendradhata, M.Sc, Ph.D, FRSPH. So he will give the speech from Swiss. Is it right? Okay. In 30 minutes. Okay. Yep. Hello, Prof. Yodi Mahendradhata.
Hello.
Okay.
Am I audible?
Okay. So you have 30 minutes to give the speech, the lecture. So the time is yours.
Okay, before I start, can the organizer share the screen of my presentation? Okay, great. Oh, okay. Okay, thank you. So good afternoon, everyone. Good morning from my side. I'm currently in Geneva. It's 7:07 in Geneva. And yeah, our friends from the Netherlands said the Netherlands is 10 degrees. Here in Geneva now it's 8 degrees, so slightly a bit colder. The sun is just about to rise, and in about 25 minutes or so, I have to make my way to WHO for a meeting. So I've been asked to deliver a brief keynote on the role of higher education institutions in advancing personalized healthcare. Next slide. Now, since I'm in Geneva and I'm going to be leaving the WHO in a few minutes, so I would like to start to set the stage a bit on what is WHO's view on personalized healthcare or precision medicine, whichever you prefer as a term. Now, unfortunately, until now, at least to my knowledge, the World Health Organization has not provided any official statement or stance on precision medicine. It has not published any specific documents or guidelines that outline a comprehensive view on precision medicine or personalized healthcare. However, the World Health Organization has recognized the importance of precision medicine or personalized healthcare in the form of reports and publication related to some elements, for example, genomics and digital health, which we know are important for precision medicine. Genomic is a key component of precision medicine. The information obtained from genomic analysis provides critical insights into individuals' genetic variations, which can influence response to treatment, disease susceptibility, and overall health. So, genomic data, healthcare providers can identify genetic mutations, variants, or markers relevant to specific disease or conditions. Digital health, as we know, is interconnected with precision medicines. Digital health technology provide infrastructure for data collection, management, and real-time monitoring, which are crucial for the practice of precision medicine. So the integration of digital health and precision medicine allows for a more patient-centric and data-driven approach to healthcare, ultimately improving patient outcome. Now, again, WHO has recognized the importance of genomic and digital health in various reports and publications. Next slide. So while here are, for example, some of the references, I mean, these references, again, do not provide an official stance. They offer at least insights into WHO perspective on some elements of precision medicine. So we're going to have a look briefly into these key references. Next slide. So let's start with the digital health. So if you look at into these documents on the global strategy of digital health, it basically acknowledged that institutionalization of digital health in the national health system requires a decision and commitment by countries because it's quite an effort. It's not something which we can do without sufficient resources or preparation. So it requires really a high level commitment. And also it requires an integrated strategy. And also WHO here encourage all ministries of health to promote the appropriate use of digital technologies for health. And also WHO here recognize the urgent need to address the major impediments faced by least developed countries, low middle income countries implementing digital health technology. So those are the key points which we can derive from this global strategy, which at the least highlights WHO's acknowledgement, recognition of the importance of digital health as part of precision medicine, and that it's something that they are pushing for. Next slide. Now, this is the genomic parts, and this is actually interesting because this report is over now 20 years. This was published in 2003. So not something new. So WHO has kept their eye on genomics for almost 2 decades now with this Genomic and World Health Report. And here are some of the key points takeaway from this report. So for example, that here they highlight that benefits from genomic research will not benefit countries which do not have a functioning healthcare system. So there's a risk of disparity, inequity across the world if you have advanced in genomic research, but there are some countries which are lacking in terms of functional healthcare system. And also that advances in genomic must be assessed for relative value in the practice and delivery of healthcare compared to the cost and efficacy of current approaches. So it doesn't mean that it could replace everything, but you have to really compare the efficacy of current approaches to public health, disease control, and provision of basic preventive medicine and medical care. And also that the existing conventional tried effective approach must not be neglected while we still also explore the medical potentials of genomic. Next slide. The potential medical application of genomics we know are so considerable, but it's also difficult to actually predict when can we actually see major advances in terms of impact in clinical practices, because it's something which remains to be seen because the application is still quite limited in terms of scale. And then partnership between developed and developing countries is critical, and that will take the field forward. And also it's time actually for quite a while now to come to plan to fair distribution of DNA technologies and potential clinical benefits to ensure, and this is important, that genomic does not widen the gap between rich and poor countries of the world, because that's a high risk that if we don't take care of this, then what we will see is that there will be wider and wider gap between rich countries and poor countries due to advancement of genomics. Next slide. And then we need a coherent policy framework to ensure that DNA patenting stimulates science economic progress by strengthening contribution of global research community for the health problems also in developing countries. So to make sure that the benefits also goes to developing countries. All forms of recombinant DNA technologies actually raise important safety issues, so that requires careful monitoring and control. And all societies, all sectors, including politicians, health professionals, educators, and public, need to be educated about the fundamental principles of genetic research, its risk, and ethical issue. So those are the 9 key takeaways of this Genomic and World Health. You can download it and see its full report, but those are the 9 key takeaways of this official report from WHO. Next slide. And this is the newest report that they have about accelerating access to genomics for global health. And here there are 4 key takeaway messages here, 4 key recommendations, in which the recommendation is to promote the adoption or expanded use of genomic in all countries. And number 2 is important, identify, overcome practical issues that impede the implementation of genomic. And number 3, foster collaborative activities to promote all aspects to advance genomic, because you cannot do this without collaborations. And number 4, last but not least, promote ethical, legal, and equitable use and responsible sharing of information obtained with genomic method through effective oversight, rules, and standard in the practice of genomic. So this is the latest one. This is a 2022 document, so it's just last year. Next slide. And more recently, due to all of this important development, WHO has already convened a technical advisory group on genomic, which they've met the first time a couple of weeks ago in October. And now they're set out to be able to safeguard the development of genomic for global health through their position as technical advisory group on genomic. Next slide. So that's just to set the stage of what's going on at the global stage in terms of precision medicine, personalized healthcare. Again, there is still not yet any official stance of the WHO on personalized healthcare or precision medicine, But you see there are some elements which have been under the radar of WHO for quite a while, and there are some issues that they're concerned with, and they're actually promoting genomics digital health because they see the potential benefits, but they want to make sure they safeguard the risk, and most importantly, that we do not disregard the risk in terms of equity, of disparity between rich and poor through technology. So within that context, what are the roles of universities in advancing personalized healthcare? Universities like Gajah Mada or any universities in the world, be it in high income, middle income, or low income countries. Next slide. So this is, in summary, 8 roles, which I think is key roles of universities in advancing personalized healthcare. So first of all, of course, research and innovations, and then genomic sequencing and analysis. And then you have educational programs, then you have interdisciplinary collaborations, clinical trials and translational research. You have ethical and legal considerations, you have community engagement, and you have collaborations with industry and government. Now let's unpack this box one by one. Next slide. So in terms of research and innovations, now many universities are hubs of scientific research and innovations in many fields. So universities conduct cutting-edge research in genomic, molecular biology, bioinformatics, and other fields, which could advance understanding of the genetics and molecular underpinnings of diseases. And even as we speak, and already for decades, researchers at universities like ours explore genetic variations, biomarkers, and disease mechanism, which paved the way for development of personalized treatments and intervention. So that's what we mean by the role of universities in terms of research and innovation. Next slide. And then in terms of genomic sequencing and analysis, many universities, including ours, we house state-of-the-art genomic sequencing facilities and bioinformatics centers. And so these facilities, these centers enable high throughput sequencing of genomes and analysis of complex genetic data. And university-based genome centers certainly contribute to the generation and interpretation of genomic information, which is critical for precision medicine. Next slide. And then in terms of educational program, one of the key functions of universities is, of course, teaching, education. So universities, some offer specialized educational programs in genomics, genetic, bioinformatics, and related disciplines. So these programs train the next generation of scientists, clinicians, and healthcare professionals in the principles and practices of precision medicine. And this program could include undergraduate, postgraduate, as well as continuing education and professional development courses. So as you can see there are a lot which can be done through the educational program to pave the way for the future of precision medicine. Next slide. And then, of course, interdisciplinary collaborations is something which is critical because we know precision medicine requires interdisciplinary collaboration. It requires collaborations among diverse experts, geneticists, biologists, clinicians, data scientists, and ethicists. And universities already been doing it for quite a while, facilitating interdisciplinary collaborations, fostering exchange of ideas and expertise. So we have the capacity to do that. And many universities establish interdisciplinary research centers and institutes dedicated to precision medicine. In our case, we have a special working group on genetics, for example. Next slide. Now, clinical trials, translational research, this is also very important to bridge between research into practice. And many universities like ours have affiliated medical centers or teaching hospitals where clinical trials and translational research for precision medicine can take place. And if we have these are trials which can test the effectiveness and safety of personalized treatments and interventions. So these academic medical centers, which are affiliated to universities, are essential for bridging the gap between research discoveries and clinical practice. Next slide. And then also very important, ethical and legal considerations, because it's paramount in precision medicines. So universities do conduct research and provide guidance on ethical issues related to genetic testing, data privacy, informed consent, and equitable access to precision medicine. We have research ethics committees and experts in universities which play a crucial role in ensuring that precision medicine practices and research adhere to ethical standards. And universities also can contribute to the development of policies and guidelines that govern the responsible use of genomic data. Next slide. And then also, this is often forgotten in the field of personalized healthcare or precision medicine, the importance of community engagement, community and patient engagement to be exact. So universities can engage with broader community to raise awareness of precision medicine and its potential benefits. We can offer public education programs, and we can also participate in community health initiative and collaborate with local healthcare providers to bridge between the development of precision medicine and what's happening in the ground with the community. So community involvement can help ensure that underserved populations have access to the benefits of precision medicine, and that the approach is implemented equitably, which is again, we need to stress over and over again. We don't want to push or drive technologies which widen disparity between the rich and the poor. So this element of community engagement is very important. Next slide. And then last but not least, collaboration with industry and government. Universities often collaborate, and we do that often, collaboration with pharmaceutical companies, biotech firms, government agencies, nonprofit organizations, which all aims to accelerate R&D and implementation. And this can be applied for precision medicine, because such partnership can lead to innovations, clinical trials, and translations of research finding of precision medicine into day-to-day practical applications. Next slide. So in summary, those are the 8 key roles of universities in advancing personalized healthcare: R&I, research and innovations, genomic sequencing and analysis, educational programs, interdisciplinary collaborations, clinical trials and translational research, ethical and legal considerations, community engagement, collaborate with industry and government. So in a nutshell, universities are central to the development of precision medicine through our research, education, clinical practice, and collaborative fraud efforts. And universities, we can drive progress, we can produce knowledge, and we can train the workforce needed to usher in a new era of healthcare that is personalized, data-driven, patient-centric, and equitable. Of course, this is much easier said than done. It takes quite a lot of efforts, it takes quite a lot of resources, time, and energy. It's a long journey, but it's a journey that we need to take despite all of the challenges, again, for the sake of better health, a more equitable health for the world. Next slide. Okay, I think that's it. I apologize that I won't be able to stay for long because I have to catch my bus to the World Health Organization. But this is my email address. So if any of you are interested to discuss further or would like to pose any questions, feel free to email me. Of course, I cannot promise I will respond immediately, but I will get back to you at the soonest whenever I can. That's all. Thank you for your attention. Thank you very much. Enjoy the rest of the summer course.
Okay. Thank you very much, Prof. Yodi, for the talk about the role of the higher education for the implementation in precision medicine. Thank you so much, Prof. Yodi. Hope you all of your activities in Geneva goes well. Thank you. Good afternoon. Okay, so give applause again for Prof. Yodi. Okay. And then we will start our session. We will talk about the pharmacogenomics, and it will be start with the presentation from Dr. Ruzilawati. Yes, she is from Universiti Sains Malaya, Malaysia. So I would like to invite her to come to the stage. Okay. So, Dr. Ruzila, you have 30 minutes for your lecture. Okay.
Okay, thank you, Ibu Tia. Okay. Okay, Assalamualaikum warahmatullahi taala wabarakatuh, and and good afternoon. Okay. Okay. So I am Dr. Ruzilawati Abu Bakar from Department of Pharmacology, School of Medical Sciences, Universiti Sains Malaysia, Kelantan, Malaysia. So, so I would like to thank summer course community, the community of summer course, for inviting me to share our experience in conducting research in genetic polymorphisms, the association between genetic polymorphisms and addictions. Okay, so before I go further, I would like to no. Not this slide. Wrong slide. Okay. Okay, so let me introduce you our School of Medical Sciences, Universiti Sains Malaysia. Okay, USM or PPS, School of Medical Sciences, USM. We located in Kota Baru, Kelantan, the east coast of Malaysia. Okay, pointer. Okay, so I need to take 1 hour flight from Kota Baru, Kelantan to go to Kuala Lumpur. Pointer is not working. Sorry. Okay. So I need to fly from Kota Baru to Kuala Lumpur. Okay. And this is our School of Medical Sciences, Universiti Sains Malaysia, or Pusat Pengajian Sains Perubatan. Okay, so in PPS, School of Medical Sciences, we have undergraduate studies or undergraduate programs as well as postgraduate programs. Okay, so Okay, so this is outline of my presentation today. Okay, for introduction, what is genetic polymorphisms? Okay, so genetic polymorphisms, they refers to the presence of 2 or more variant forms of specific DNA sequence that can occur among different individuals or among different populations. Okay, so or in other words, polymorphisms can detect slight variation at the level of DNA. So we also have protein or enzyme polymorphisms. Okay, So there are types of genetic polymorphisms. So we have single nucleotide polymorphisms, okay, or SNP, variable number of tandem repeats, simple sequence repeats or simple tandem repeats, and we also have restriction fragment length polymorphisms or RFLP. Okay, so in our research we done we done research on nicotine addiction, which includes smoking addiction and also illicit drug addiction, which include amphetamine type stimulant or ATS and opioids. Okay, so as we all know, nicotine is a major substance in the tobacco, and nicotine addiction is nicotine is responsible for addiction in tobacco product, including cigarette smoking and also vaping or smokeless tobacco. Okay, so when someone smoke, nicotine will enter to our body, and this nicotine will spread throughout the body, and the nicotine will enter the outer membrane, capillaries, and cross blood-brain barrier in the brain, and then nicotine will bind to the receptor on the brain cell, and then they will induce the release of neurotransmitter, for example, dopamine, serotonin, or serotonin. And then this neurotransmitter will lead to the addiction. Okay. So nicotine addictions occurs because nicotine activates the brain's reward system. Okay, so this lead to the release of neurotransmitter like dopamine, which create feelings of pleasure and reinforce the behavior of using tobacco. Okay, so over the time, the brain becomes familiar to the presence of nicotine, and the smokers will develop a tolerance, meaning that they require more nicotine to achieve the same pleasurable effects. So this can lead to dependence and addiction. Okay. So there are genes that can be considered as candidate genes that are associated with nicotine addiction. Okay. So namely we have nicotinic acetylcholine receptor, okay, and nicotinic acetylcholine receptor genes. We have genes that involve in nicotine metabolisms, for example, cytochrome P452 A6, and we have dopamine receptor, D1, D2, D3, D4 genes, and also we have dopamine transporter genes, serotonin receptor, and serotonin transporter genes. Okay. So our research groups, we perform the research in order to estimate the prevalence of nicotinic acetylcholine receptor, serotonin receptor and transporter, and dopamine receptor and transporter genetic polymorphisms associated with a smoking behavior among Malay smoker and non-smoker. Okay, and we also try to determine the association between these gene polymorphisms with smoking behavior among Malay smokers. Okay, so this is the flow chart of our study. Okay, we recruit 224 smokers and 248 non-smokers. Okay, and this study was approved by our committees, our ethics committee members, School of Medical Science, and the work has been carried out in accordance with a declaration of Helsinki for experiments in humans. Okay, so we take 3 ml blood, and then we extract DNA extraction, and we perform PCR FLP, and we confirm the genotype by DNA sequencing. Okay, for non-smoker, we assess them with a smoking behavior assessment, and also with a progesterone test for nicotine dependence, or FATND. Okay, so this test is used to provide measure of nicotine dependence related to cigarette smoking. Okay, so a little bit on nicotine acetylcholine receptor. So when nicotine exerts its pharmacological and physiological effects in our body by binding to nicotine acetylcholine receptors. Okay, so molecular analysis, they have identified the expression of 9 subunits of nicotine acetylcholine receptor in our central nervous system. We have alpha 1 until alpha 10. Okay, so for alpha in our study, we choose alpha 4 subunit nicotine acetylcholine receptor because from previous study, they considered that this alpha 4 subunit nicotine acetylcholine receptor can be considered as a good candidate genes for smoking behavior. Okay. So we perform genotyping of alpha 4 by using PCR and RFLP analysis. Okay. So from the result, we found that there was no significant difference were found in genotypes, and also no significant difference in allele frequency in both group, smoker and non-smoker. Okay, so for association of nicotine acetylcholine receptor genotype with progesterone test nicotine dependence score in smoker group, we also find no significant difference. Okay, so we can conclude that our research in nicotine acetylcholine receptor genes, there are no significant association were observed between nicotine acetylcholine receptor with smoking and non-smoker groups. Okay, and this mutation in alpha 4 genes are rare among Malay populations. Okay, so we have successfully published our research in Egyptian Journal of Medical Human Genetics. Okay, this is Scopus Index. Okay, next we go to serotonin. Serotonin is also one of neurotransmitter that derive from amino acid tryptophan in our brain. Okay, so polymorphisms that affect the serotonin system, such as serotonin receptor and serotonin transporter genes, have shown that they are linked between this gene with smoking behavior, because nicotine increase serotonin release. So variation in the serotonin gene system may influence some aspect of smoking. Okay, for example, mood variation during nicotine withdrawal, and also they have linked to smoking behavior. Okay. So a little bit on serotonin receptor, we have 5-HT receptor. They can be classified into 7 groups, and one of them is subgroup 5-HT2. So this 5-HT2, we can divide into further into 3 subclasses. We have 2A, 2B, and 2C. So for our research, we concentrate on 5-HT2A genes. Okay. So these 5A2A genes, from the previous study, they found that these genes are associated with emotional disorder, and also associated with alcohol dependence, and also related to smoking behavior. Okay, so indeed, the polymorphisms in serotonin receptor has been associated with continuation of smoking behavior, and also in the initiation of smoking behavior. Okay, so we perform genotyping by doing PCR RFLP, and further confirm with DNA sequencing analysis. Okay, and then again from the result, we get that there were no significant difference in genotypes and allele frequency in both group of smoker and non-smoker. Okay, and also in association study with FTND score also was no significant difference were found. Okay, so we try to compare our study with association study between different ethnic groups. Okay, so from their study, they found that in study in Caucasian Australian, okay, they found that those who have TT genotype are associated with smoking behavior. And in Brazil population, they found that those who have CC genotype are associated with smoking behavior. Okay. So we also did our research on serotonin transporter, okay, 5-HTT. So this 5-HTT genes is located in the long arm of chromosome, and the serotonin transporter is control the duration and concentration of serotonin neurotransmission in the synaptic cleft. Okay. So this serotonin transporter genes has been, from previous study, they found that these genes has been linked to psychological traits and also related to nicotine dependence. Okay, so they found that in 44 base pair insertion and deletion polymorphisms located in serotonin transporter link polymorphic regions was identified within this gene is they resulting in 2 allelic variants. We have long and short alleles. Okay, so these 2 alleles alter the transcriptional efficiency of 5-HTT genes. Okay, so we perform PCR analysis. Okay, and confirm with DNA sequencing analysis. Okay, so for this serotonin transporter genes, we also found that there were no significant difference in genotype and also in allele frequency in both group. Okay, so we compare our study with other study. Okay, so in our study, we established that this serotonin transporter polymorphism was not associated with smoking behavior in our Malay population in Malaysia, and this finding is consistent with 5 previous studies in Dutch, American, Austrian, Polish, Greek, and Thai population, meaning that they have no significant association between serotonin transporter gene polymorphisms with smoking behavior. So, in Japanese population in China and Sweden, they found that L-allele, those who have L-allele, was more likely to be carried out by smoker compared to non-smoker, and those who have L-allele were found to have higher intensity in smoking behavior, and they are more likely to be dependent on nicotine. Okay, so in USA population and Italian, they found that smokers with homozygous and heterozygous S-allele were able to quit smoking more easily. Okay. So we also publish our result in this journal, okay? And then we go to dopamine, okay, and nicotine. Dopamine, nicotine, when they enters our body, they will cause the release of dopamine in the brain, in the area of nucleus accumbens and ventral tegmental area. So this nicotine will continuously exert reward effects by increasing the production of dopamine in the brain. Okay, so the release of dopamine within this area will mediate the rewarding and pleasurable effects with, which is a critical mechanism for initiation and maintenance in nicotine addiction. So nicotine, from previous study, they found that nicotine addiction has been significantly associated with polymorphisms in the dopamine receptor and also in dopamine transporter genes, which they play roles in dopamine pathway. Okay. So this a little bit on dopamine receptor. Dopamine receptor are rhodopsin-like 7 transmembrane receptor or G protein couple receptor. Okay. So dopamine receptor can be divided into 2 D1-like receptor and further divided into D1 and D5, and we have dopamine 2-like receptor, okay, can divide it in, further into D2, D3 and D4. Okay, So dopamine receptor genes has been reported to be one of the plausible candidate genes for nicotine dependence. So in our study, we choose three genes, D1, D2, and D3. Okay. And for dopamine transporter, we choose D80 gene or SLC6A3 to study the association between gene polymorphisms and smoking behavior. We use PCR-FLP for genotyping for the dopamine transporter and also dopamine receptor for D1, D2, and D3. Okay, so in our result, we found that in DRD1 there was a significant difference in genotype and also in allele frequency. Okay, and we have found significant difference in genotype in DR2 and also DRD3 genes. Okay, so because of this significant difference, we further analyze using multiple regression analysis. Okay, so we found that for AG genotype in DRD1, they have prevalence higher in smokers, meaning that they have association between genes and smoking behavior. Okay. For the RD2, for LLA1, A2 genes, they also associated with smoking behavior. And for DRD3 in CCLs, we found that these alleles are protective genotype. Okay, so from our findings, we can conclude that genetic polymorphisms in dopamine receptor D1 and D2 was significantly associated with smoking status, manifest that increased risk of predisposition to smoking behavior among our Malay male population, and we found protective effect of the DRD3 gene to smoking behavior. And for the dopamine transporter, there was no association between this gene polymorphism with smoking behavior we observed. So again, we published our study in Q2 journals. Okay, okay. So for second research, we done our research in drug addiction, particularly in ATS, amphetamine type stimulant, and opioid. We try to associate with gene polymorphisms. So a little bit on drug abuse. Drug abuse is the chronic disease which involve the compulsive seeking and drug use that can be influenced by both genetic and environmental factors. So in Malaysia, opioid and ATS are the main drugs that had been abused among our drug users. Okay, so opioid, predominantly heroin and morphine, they can produce morphine-like effect or also can produce euphoria characteristic. Therefore, many person use this for abuse. Okay, for amphetamine type stimulant or ATS, can be categorized by stimulants. Okay, so ATS is the second most commonly used as an illicit drug type and is more widely used than compared to heroin. So for drug addiction, they again is influenced by both genetic and environmental factors. So in our research, we choose dopaminergic genes that involve in dopaminergic pathway. Okay, so because from previous study, they report that these genes are involved in drug addictions or can be candidate genes. So for methodology, we recruit 70 male subjects drug abuse that they take meets amphetamine type stimulant and opioid dependence. So we enroll from Methadone Clinic, Hospital Universiti Sains Malaysia. So for drug dependent subject, we were diagnosed, we use DSM-5 and they must fulfill the criteria for ATS and opioid dependence. And to collect the demographic and clinical data from the subject, we use addiction severity index. Okay, so we genotype the subject with dopamine D2 receptor, dopamine transporter, and dopamine beta hydrolase genes. And the result, we found that there was significant association between DRD2, dopamine beta hydrolase and dopamine receptor, gene polymorphisms with mixed amphetamine type stimulant and opioid dependence at the genotype and allele levels. However, no association were established in dopamine transporter. Okay, so this is we also try to compare our study with other different ethnics. Okay. So again, we publish our research in this journal. Okay, so for take-home message, the study of the association between genetics and smoking behavior and as well as in drug addictions are complex. Okay, because they depend on many factors. Okay, making that the association are unique to each populations. So any findings of association between genes, polymorphisms, and smoking behavior as well as drug addiction may only be true for the population study. Okay, so this is an example of pharmacogenetics or pharmacogenomic research that can bring forward to personalized medicine, okay, or to precision medicine to the addiction field. And we can use our result or this result to identify the best form of treatment and prevention in addiction. Okay, so again, when we know the genotype of the smoker, we can use this genotype profile to precision medicine. Okay, let's say if patient want to quit smoking, for example, anybody know what drugs can be used for quit smoking? What drugs can be used for quit smoking? Anybody? Have you heard about varenicline? And so we can use, let's say, we can use, as I mentioned before, that those who have serotonin transporter, who have S allele is easy to quit smoking. Those who have S allele, easy to quit smoking. Therefore, to interpret or to implement personalized medicine in this addiction, maybe example, we can give those who have S allele, maybe we can give a low dose of varenicline for this patient or these smokers. Okay. So again, special thanks to my postgraduate students and collaborators and also grants. So with that, thank you.
Dr. Ruzilawati, maybe you can have a seat here. Okay, yeah. Please give applause to Dr. Ruzilawati again. So from her presentation, we already know that the changes or the genes polymorphism has the association with the smoking behavior. So as the previous speaker said before, that the bioinformatics has the important role in the implementation of the precision medicine. So the next session will be Dr. Piroon Jenjaroenpun from Mahidol University, Thailand. So he will explain to us about how the bioinformatics take part in the implementation of clinical pharmacogenomics. So, Dr. Piroon, give applause to him.
Hi everyone. So thank you organizer that invite me to speak here and having me here today. So today, actually, we have both experimental and also bioinformatics. So how many of you know the word bioinformatics? How many? Few? Not a lot. So what is the bioinformatics does? So actually, bioinformatician, previously, my bachelor degree, actually, I study microbiology. Yeah, and then I feel that when I work in the lab and fail in the lab, so don't like it. So that's why I changed my field so into like bioinformatics. Mostly, I do programming, right? And you can fail and you can rerun again and again. So that's good. So, but yeah, once I know bioinformatics, so I study bioinformatics and in master degree and also in Ph.D. So, but today, so it will be combined between experimental in the pharmacogenomics and the bioinformatics. Today, this is the case that I'm gonna give you when we use bioinformatics and new approach, nanopore sequencing approach. So how many of you know about nanopore sequencing? Nanopore sequencing? No, so it's very new. And how many of you know about next generation sequencing? Raise your hand. Next generation sequencing. So you know, okay? So next generation sequencing is the technology that you can define, like you can identify the sequence of DNA or RNA. So, and that we can use as the application to study things. So today we talk about HLA class 1 typing using that nanopore sequencing. And it gives us the high resolution to identify the genotype of the HLA. So this is my lab. So it's very new lab. We start the lab 2021. And the name is Serialized Long Read Lab. Actually, doing only long read? No. We do both long read and next generation sequencing. So, and you can see that we have mix between experimental scientists and bioinformatics. So we have like 1, 2, 3, no. So 1, 2, 3, 4, 5 bioinformatician. And 1, 2, 3, another 4 just coming. So that mixture between the person who do the lab and the person who do bioinformatics. Why? We have to talk to each other when we get the result from the next generation sequencing or from the sequencing approach technology. So once we have the data and we have to analyze the data, that's my job and my team that do bioinformatics. So yeah, and you can see that is the mixture between them. Now, back to the case study that we have today is human leukocyte antigen set data gene, HLA gene. So it's very important related to immune system. So it will know whether you have the new protein or new organism inside your body or is your own cell. So it can discriminate that kind of thing. And it's very important. So if anything like pathogen go to your body, so your immune system know this is the new one and you have to kill it. So, and this one actually belong to the chromosome 6 of human. And then that region, we have the region of the M.Sc, you have many genes related to HLA, A, B, C, DR, DQ, P1, DP. So it's actually clustered into like class 1, class 2, and class 3 here. But today, we're gonna focus only in the class 1, ABC, easy. Okay, in class 1, ABC, each gene, you're gonna have this structure. So, do you know about exon, intron? Yes, okay, that's good, easy. So you have the exon here, and this exon actually express, right, transcribe into RNA, and RNA can translate into what? To protein. So this region actually gives you, this is the gene, and you get the RNA of it. And then the exon here, the red one, gonna concatenate to each other and translate into the protein. So, and that protein can make a structure. This is the structure of the HLA. So, and then the code that you see here is form a structure. You can see the exon 2 is alpha 1, exon 3 is alpha 2. Both of them actually belong to the group of the HLA, and that can present the antigen or do something. So, and this important region. So therefore, some technique they use typing of these two regions. Okay, so that's the background of the gene. And this gene actually quite diverse. So it have many forms. So this is the class 1 only, ABC, and you have different pattern of the sequence of the HLA. Each person here may have different type of the HLA. Totally, we have like 21,000 pattern, but it actually is more than that because this is old data. It's old data, maybe 3 years ago. So, and each gene, you have 6,000, 7,000 of the pattern. So, what is the pattern that I mentioned too? The pattern is actually, they study the form of the genes, whether they have chain of the amino acid, whether they have chain of the coding on the nucleotide in the exon. So, and then they give a name. They give a name. So this is HLA-A, this guy. HLA-A, you have the name of the HLA. This is the pattern, or you so-called allele. So each one here have this kind of allele. So the allele here, if you have gene A, HLA-A, so you're gonna have HLA-A. If you have B, you're gonna have HLA-B. C, HLA-C. So all of you have both of them. All of you have ABC. So this is for A only. So this A, if they have the form of the structure, as I showed you just now, this one, oops, this one. If they have the form of the structure that work with some group of the antigen, so if you have like the first one allele group that can bind to things, and then if you have the second one here, second one here indicate that the protein is the same protein. So different type of the HLA have the same protein. So I have the example in the next slide. So just say that if you have the digit the same, 0, 1, and your friend have 0, 1, 0, 1 here, you should have the same protein. And then the third field is the exon synonymous mutation. So it means that your nucleotide changed, but protein doesn't change. Yeah, so that additional. And then the last one is the intergenic region or intron, which is outside the coding region. So the last one is suffix. If you have like no protein developed, so it will be N. For example, this is the case example of the what I term. So HLA-A, you have HLA-A, all of them HLA-A, they have the HLA-A. All of them have 0, 1, the same group of the HLA, and 3 of them here in the second field have 0, 1, 0, 1, 0, 1. All of them have the same structure of the protein. Yeah, because the sequence is exactly the same. And this guy have 0, 2. It means that this protein and this protein have a little bit different. So there are some amino acid different. And then the last one here is no protein because you have N here. So the last field, the third field here, even you have different like 0, 1, 0, 2 here, they have the same protein anyway. Okay, they have the same protein anyway. It just indicate that you have some different in the nucleotide, but the protein is fine. All right, so then why you have to do HLA study? Why you have to do typing? Because as you see, is many type of the HLA, right? Many kind of the allele. So if you study on that, so it's in many field. It's in many field. You can study related to like autoimmune disease, cancer, infectious disease, and also you can use in terms of the protect the rejection of the transplant organ. So to check whether the organ is fit to patient or not, for example. And also the last one here is the link to the adverse drug reaction. So which is make you feel bad when you get the drug reaction. So, and actually Professor Chonlaphat have example and picture that you can see how like, what is that term? How dangerous of that kind of drug reaction. Okay, so this is case example. This is just one example that I have picture here, but he have a lot of picture. So HLA related to pharmacogenomics or pharmacogenetics. So it's actually related to adverse drug reaction or ADRs. So this is the example when you have HLA-B, so that gene B, right? HLA-B, and then 1502. So if you have this kind of protein, then you take this drug, carbamazepine. So you're gonna have this kind of symptom, yeah, because of that adverse drug effect from that allele with the drug. So and this is the symptom that you can get if you like have fit of the HLA type that adverse with the drug. So this is the study in Thai population, again from Professor Chonlaphat. So we have known that if we check related to carbamazepine, you have 1502, which is related to that adverse in carbamazepine. And also this one is about 14%, 16%. But that is not only drug that make the symptom. You have other kind of drug here as well. So that you have to check whether you have the HLA type fit to the drug and then get the adverse drug. So, and in Thailand, so we have the implement of testing the HLA when you're gonna get the carbamazepine or allopurinol. So in some certain case, we will check whether you have HLA-B 1502 or you have HLA-B 5801. If you have that, you should not take it. You can't take it. Otherwise, you will have the symptom. So, and yeah, is that for Thai people? It's a lot, 15 or 16%. But in the same drug affect other kind of the population, like Japan, European. So if you have HLA-A 3101, so you could get the drug reaction like Thai people, yeah. So and this is allopurinol for Thai people. So it means that population independent. So it's not like carbamazepine will fit only 1502. No. So from other population, you have to check what is the allele or what is the HLA type for that population that you have. So, and therefore, if you test only 1502, is that enough for the checking whether you take the carbamazepine? No. So, because the new study show that even you don't have this 1502, you still get the adverse drug effects. So, therefore, there are some additional study more and more, and then give you like 1508, 1511, 1521. So all of that suffer with the drug effect as well. And yeah, some related to HLA-A 3303. So therefore, it's maybe if you want to take the carbamazepine, at least 6 allele should take or takes it. Okay, so now, the HLA type is actually, every one of us have 2 allele, right? One from your father, another from your mother. So when you have the zygote mix, right? So you're gonna take one allele or one chromosome from father. For example, this one is HLA-A0207. So go to that baby. And then mother give the HLA-A1101 to this baby. So, and finally, you're gonna get 2 allele. So one person have 2 allele, right? So you have this allele 1, 0207 and 1101. So therefore, when you test it, it's not just one allele, right? You test 2 allele and check whether you have what kind of the allele type. So this is the technique of the typing, HLA typing by sequencing. So actually they have more than this technique. So this one is one of them, sequence-based typing. So you have the primer set and identify the sequence during the alpha 1, alpha 2, which is related to the growth of the HLA structure. And then this is the previous technique. And then we have next generation sequencing come. So this guy, you can sequence the whole thing, the whole genes, and then see very high resolution. So you compare with that. If you typing this, you have like multiple type possible, but this next generation sequencing, you have just one. So after you sequence, you see what kind of the type you have. So, and this is how we use it, right? Next generation sequencing, when you have genome, this is your genome. You have one allele from father, one allele from mother. And then once you have your DNA, we extract DNA, chop it into the small size, like that. And then when we want to do typing, we have to do assembly. Assembly means that you combine all the information here if they are overlap. If they are overlap here, we just concatenate them, right? And make the as long as possible sequence. And in this case, you have long sequence, but you have problem with this region, because this region for mother and father have different, and then you don't know whether this one go with which one. So because your sequence is so short, therefore you're not able to make a link between this GA and GC or AC or GT. So it's impossible for that if they are far away. But if you have long read sequencing, so you have the sequence that long enough to cover the genotype from the beginning at the end of the genes. So that means you can have the sequence that resolve, and you know that GA will go to AC always, and then AT go to GT always. That can reproduce from, like reconstruct the allele from the DNA that you have. So now, Oxford nanopore sequencing. So this is the technology that can help to do the long read sequencing. So it's actually the palm size of the machine. So you can buy, this is $1,000 US dollar. You can buy this guy and the full cell and do sequencing. It's not that expensive compared to next generation sequencing, because that's really expensive. So, and then within the full cell here, you have a lot of nanopore. So why it's called nanopore sequencing? So you have a lot of nanopore. And then this nanopore is gonna have the ion. When you do sequencing, ion will flow to the pore. And then this flow, you can calculate, you can detect how many ion go to the pore. And if you have nothing to block the pore, the ion will go through the pore equally. So therefore you have like straight line. This is the plot of the number of the ion. So if you have same amount of the ion, it go to the straight line. But if you have the molecule block the pore, what happens? The ion will be lower, and then it's gonna drop the graph, right? And that means you can use a lot of the DNA, right? And it generate the pattern like that. So multiple DNA make a pattern, and that pattern can change into ATCG by machine learning. So, and that nanopore use this approach to define what is the base. Okay, so this is what happened inside the nanopore. So the DNA with the adapter will go to the pore over here. And then doing that, we have ion will go to the pore. Now DNA go into the socket here, and it's block the current signal, and it's drop. And it's drop as the pattern because of different base change. When you move one base, the base is changed. You have different pattern, and it's go along the way. So yeah, that's how nanopore do sequencing. And we use that technology to study the HLA typing. Yeah, and we just published this year. This is the design of the study. So we start with testing whether this technique is work or not. So we buy the DNA from US, that's standard DNA, that we know what is the HLA type already. So, and that 11 sample. So we take that, and then we will use the different set of primer, and also use different set of the library preparation of the nanopore. And then we use the bioinformatics to solve that. Okay, so this is what we test. We actually test the ligation kit and rapid kit. Nanopore provide you the two way of the library preparation. You can add adapter at the end of the DNA. So this is DNA, right? When you have the DNA, you attach the adapter at the end, to end of the DNA, double strand. And then you get the adapter at the end of it. But for the rapid kit, is insert the adapter inside the DNA. It's just bind to the DNA and cut, make a cut, and then add adapter. So that's the way of these two different. And this one is much faster compared to this guy. Okay, now analysis. After you run nanopore sequencing here, what you will have is DNA sequence. So that the sequencing do, right? You have DNA sequence, you have very long sequence of the DNA, and then you have multiple of them, probably million of them. And then what you do is that you have to have the program, so you write the program. To analyze them, first, you have to check quality, whether the quality of the sequence here is good or not. If it's not good, throw away. If it's good, keep it, okay? After that, we do read collection. So you combine the read that have very similar sequence and then make a collection. So after that, after the collection of the reads, then we group the read based on the allele. So with this, we try to find where is the sequence that have the similar pattern, similar sequence. And then we separate them. And this is the program, right? After separate them, then we have to set allele 1 and allele 2 separate group. After that, we combine and make one assembly of the read here. So if they have overlap, so we make an overlap and then concatenate them into one sequence. So now we have the sequence, which could be one allele and two allele. And then we do blast with the database of the HLA, and that make us able to annotation whether this HLA A or no, haplotype 1 is what kind of HLA. This is HLA B 1502. And another one is HLA B 5801. So based on this approach, so we can identify and we know that there are two alleles in one person. So therefore we get two alleles. All right, so then we test with the standard DNA that we buy from US. And then we see that, okay, the two set of the primer, this set we use the set from the publication. And this one we buy from commercial. So all type is the commercial that they sell the primer set to identify or to amplify the HLA genes. And then we got like 100% accuracy for the commercial one, not the free one. Okay, so all the DNA is correctly typed. So then we go to our 9 patients that we want to test. Then we use our nanopore approach and then do typing. As you see the analysis, right? We can identify HLA-A, HLA-B, and what kind of the pattern it should be in each sample. And based on the traditional way to typing is the PCR SSP. So we have 5 positive B star 5801, 1, 2, and then 1502, 1, 2, 3. So totally 5 that positive and other negative. So, and we check with our pattern that we identify and correctly identify. So we have all of them present. But since we have the pattern, then we could use that with another knowledge, right? So we go to the publication that we tell that we should test 6 of them, right? 6 allele that potential adverse drug effect for the carbamazepine. And we found that we have additional 1 sample that have 1521, which is in the list. So it means that this patient maybe need to be careful with the carbamazepine. And some other that they already have, this is not carbamazepine, this is allopurinol, another drug, but they have potential to have like drug adverse to carbamazepine as well. So, and then we go to Prof. Chonlaphat paper again and get some additional knowledge that if you have like this guy 1501, so you have chance to have drug adverse in another drug, not these 2 lists that we have in hand. So it means that once you do typing, right, you know HLA of your own, then you can check what is HLA that you have is affect which kind of drug. And that knowledge can come in the future as well. So right now we maybe not know yet, but in the future we will know. For example, then we can test your HLA type with that drug. Okay, this is the conclusion. So with our technique using the nanopore sequencing and HLA, it offers high resolution HLA typing. You can have like the pattern with the 3 field and 2 allele. Then cost effective. So even you have small lab or developing country like us can use it. So because it's not that expensive compared to next generation sequencing. So if you have like $1,000, you can buy it and then test your own genome, for example. Ethnic independent. So once you have the pattern, even you are Caucasian or you are Asian, so you can just align to the reference in the database and check whether you have like leaks to the drug or not. Rapid result is kind of so when you do the experiment, you do 1 day. So do sequencing 1 day, and then you do analysis maybe 1 or 2 days. If you have pipeline, it's very quick. It's maybe just 6 hours, you get the result. And then you can reduce the drug adverse effect. So, and accessibility, yeah, as I tell you that the cost is low compared to the NGS, normal NGS. So therefore, it's possible to buy it and use it. Okay, so that's all for me. So, and thank you for all collaborators that helped me to do that paper. Yeah, thank you very much.
Please have a seat here, Dr. Piroon. So thank you so much for your interesting presentation, because by knowing our HLA typing, we can reduce the adverse drug reaction of any particular drugs. So yeah, it's so interesting for me to know that. And how about the implementation of the pharmacogenomic in clinical setting? There will be Professor Chonlaphat from Ramatibodhi Hospital, Thailand. So please welcome Prof. Chonlaphat. Please give applause to him. Yeah. Before yes. Okay, before that, I do believe that you guys already have a lot of questions in mind, but you should keep it first after Prof. Chonlaphat and the last speaker have finished their presentation. So please, Prof, to...
Thank you so much for
your talk.
a very kind introductions. And so hi guys, good afternoon and sawadee krab. So I'm Chonlaphat Sukasem from Bangkok, Thailand. So today, it is my great honor to stand before you today to talk about the clinical implementations of the pharmacogenomics and also precision medicine. It is good for me because I'm talk after you learn about the research of the pharmacogenomics and you learn about the technique. And so right now, in this time, we are going to move forward to be the clinical implementations of the pharmacogenomics and precision medicines, and I will share you the experience how can we implement the pharmacogenetic testing in our Thai populations to prevent them from the adverse effect and maximize them to get the very high efficacy of the medicines. So actually, so this, oh, okay, it's come back. Okay, thank you. So in my talk, so I don't want you to remember the biomarkers, because actually when you practice as a practitioner, you can search and you can read in the Googles or in the database. But today, I just want you to inspire you and to know or recognize when you go to be the practitioner, like doctor, like pharmacist, dentist, or nurse, or a medical laboratory scientist. So you should be recognized that some of the patients who get like toxicity is come from their own genetic factor. And also sometimes, not just only the genetic factor of that influence to the drug response, but it might come from non-genetic factor as well. So today, I'm going to talk about the genetics and non-genetic factor that when you grow up to be like practitioner, you should keep in mind and recognize of that factor. So in my talk today, so I'm going to talk about the pharmacogenomics. So can I point that one? Okay, I'm sorry, this another side. Okay, I'm going to talk about the pharmacogenomics, and also I'm going to talk about the precision medicine. So you should be able to differentiate about the pharmacogenetics and precision medicine, because actually for the pharmacogenomic itself, it's considered just only the patient genetics only that influence to drug response, but they are not precisely enough when we predict the drug response in the patients in terms of the genetics of the patients. But also we have to comprehensive consider with the non-genetic factor of the patients, which is we call precision medicine, that consider genetics and non-genetic factor to drive the efficacy or toxicity in the patient. This is term of the precision medicine. And so in my talk, so I'm going to talk about how to imply the pharmacogenetic testing into to prevent the severe cutaneous adverse drug reactions in the patients. The severe cutaneous adverse drug reactions we can call as SCAR in this term. And also I'm going to talk about how to do the dose adjustment, optimize the dose, right drug, right time to the patients by the pharmacogenetics. And also give you an example, the clinical implementation with the standard workflow that you should be recognize that the pharmacogenetic is not just only the role of the doctor or the pharmacist only, but there are interdisciplinary approach that you should be able to work together with multidisciplinary teams. And also I'll tell you about the case study and also the future perspective of the pharmacogenomic and precision medicine that might be influence to your country as well. Okay, so before I'm going to talk about the pharmacogenomics, I'm sure that you have learned about the pharmacokinetics and pharmacodynamics already, right? So we can predict the drug response of the patients in both way, efficacy and toxicity, according to the pharmacokinetics and also pharmacodynamics in the patients. So the pharmacokinetic is a process of the body does to the drugs, absorptions, distributions, metabolism, and eliminations. So when the patient get the drug can be absorbed and circulate into the systemic organs of the patients, and also they go to the target site. And this is the pharmacodynamics. The drug, what the drug does to the body. The drug can bind to the receptor, dopamine receptor that Professor already mentioned about dopamine receptor, for example. This is a pharmacodynamics. So you may see that along the process of PK and PD, they have the function of the proteins. So therefore, in the patient who have a genetic polymorphism of the PK and PD genes, they may have alterations function of the protein in PK and PD, and they may have they may change the drug response. They may get low efficacy or treatment failures, or sometimes they may get like toxicity according to the genetic polymorphisms. This is a background of the pharmacogenetics only that we test in the clinical implementations. And so before I talk to you about the clinical implementations of the pharmacogenomics, so I give you an example of my case study. So this is my case study that the patients, the elderly patients, she is 75 years old, and she is a renal impairment functions. And we prescribe allopurinol for her to treat the condition of hyperuricemia for 1 year. She treated for allopurinol for 1 year. And so after 1 year, she came back to the hospital with the kind of the severe cutaneous adverse drug reactions. This is we call Stevens-Johnson syndrome. She came back to the hospital with the fever. She came back to the hospital with the mucosal involvement, like mouth, eyes, and also genital areas. So at the first admissions, we diagnosed her as a Stevens-Johnson syndrome because the skin detachment is less than 10%. The mortality rate is not too high. It's about 5%. So however, after 1 week, she turned to be toxic epidermal necrolysis, which is most life-threatening adverse events. The mortality rate is about 30%. And we diagnosed her as toxic epidermal necrolysis because the skin detachment is more than 30% as well. So this is the most life-threatening adverse events in the medicine. So unfortunately that we cannot help her because we did not genotype the HLA-B for her before prescriptions because she did not meet the criteria to get the national policy screening, because the national policy screening we allowed to do in gout patients, but she did not diagnose as gout patient, but she is a hyperuricemia only. So therefore she died from toxic epidermal necrolysis. So after that we have a questions because when we look into her family, so we found that her son, this guy, now taking allopurinol as well. So he asked the question that should him stop the allopurinol or he can continue with the treatments. So in this lecture, I will use the pharmacogenetics testing to answer that patient's questions, and we will come back to discuss this case again. Okay. Okay, let's go to, as I mentioned that the Stevens-Johnson syndrome and toxic epidermal necrolysis, in that case, she treated for more than 1 year because the severe cutaneous adverse drug reaction is a delayed thyroid hypersensitivity. It can be delayed until 3 months, 6 months, or 1 year. So in the past, the scar is idiosyncratic. We don't know the cause, the reasons. So therefore, it's unpredictable and it is dose independent. So however, right now, by the pharmacogenetic study, we can identify what is a predisposition factor. And we found that the genetics of the patients is a predisposition factor for the severe cutaneous adverse drug reactions. So therefore, we can predict the adverse drug reaction in the patients before prescriptions such a drug in that patient. For example, before we prescribe carbamazepine, the HLA-B1502 should be screened. For allopurinol, HLA-B5801 should be screened. And also for abacavir, which is an anti-HIV, the HLA-B5701 should be screened before prescriptions. In the patient who are the carriers of the biomarkers, even like in heterozygous or one alleles or homozygous or both alleles, we should avoid to prescribe such drug in that patients because they are very high risk of severe cutaneous adverse drug reactions. And on the other hand, in the non-carriers of the patients, the drug can be prescribed safely for them. So you may see that almost of the biomarkers is on HLA-B. So let me explain about the HLA. And actually, Dr. Piroon already mentioned about the HLA gene for you. The HLA gene is located on the chromosome number 6 at the short arms. And the HLA is a gene that codes to be major histocompatibility complex, which is a protein that is a transmembrane protein. The function is to present the antigen or to present the cell peptide for negative selections. This is a function of HLA proteins. The HLA can classify to be 3 classes: class 1, class 2, and class 3. For class 1, A, B, C is expressed on every cell that have nucleus. For class 2 is expressed on antigen-presenting cell. And class 3 is a tumor necrotic factor. So for the HLA that coding to be the M.Sc that expressed on every cell that have nucleus, keratinocyte is a cell that have nucleus. So they have class 1 presentation. And for antigen-presenting cell, they have class 1 and class 2. So when you look into the figures on your left-hand side, we demonstrate by the in silico model about the structure of the HLA, and you may see that this one is a transmembrane part, and this one is expressed above the cell membranes. And when you look into the top view, you may see the groove that can capture the cell peptide and also antigen to present to stimulate the T cell receptor, stimulate the cytotoxic T cell. So therefore, in some patients who have specific HLA, they have the structure that can capture drug molecules and present to T cell receptor to activate the cytotoxic T cell and also immune system to secrete the cytokine, ganzyme B, and also the chemotactants. And also, therefore, the cell can be destroyed by that cytokine and also ganzyme B. So you can imagine that when the keratinocyte present the antigen as a drug molecules, it can be destroyed. And so you can see the skin detachment in the patients. And in that area, so you may see that the vascular cell, endothelial cell, has been destroyed as well. So therefore, the water in the circulation leak from the vascular out from the vascular. And you may see the blister in the patients. This is a clinical manifestation that we can see. And also the vascular, the water still be leak. They have a very high pressure, so which is we call oozing. Therefore, the skin can be burst, and this is a cause of the skin detachment that you can see in the patient, originally from HLA reaction that capture the drug to present the T cell receptor. This is a role of the HLA. So therefore, the HLA is involved with the severe cutaneous adverse drug reaction, and we should screen the HLA genotyping before prescription right now. So in this slide, I summarize my study in Thai populations, and I highlighted you about the biomarker that has been implemented as a national policy screening in Thai populations. So the first one is a HLA-B1502 for carbamazepine. And so as you may look into the structure of the carbamazepine, they are look very similar structure with oxcarbazepine. So therefore, the oxcarbazepine share the alleles specific biomarker with carbamazepine, which is a HLA-B1502. In the patient who carry with HLA-B1502, not just only carbamazepine, but also oxcarbazepine should be avoided in that patient as well. And as Dr. Piroon already mentioned about the HLA-B serotype 75, which is a same structure protein, but different alleles or sequencing. So therefore, in the patient who carry with HLA-B serotype 75, such as 1521, they should avoid for carbamazepine prescriptions as well, because they have the structure that's similar with HLA-B1502 and capture carbamazepine molecule to present the T cell receptor. And moreover, in our populations, we also implement the allopurinol screening for HLA-B5801 before prescriptions of allopurinol in the patient who diagnosed as gout in our patients. And other than that, so we are now to put our effort to implement, extend our screening as a national policy screening in our Thai populations. So let me give you an example about the HLA-B1502, and I talk to you that in Thai population we found the association in this marker, but not just only for Thai population, but also the HLA-B1502 can be shown the strong association with carbamazepine induced HASN10 in Malaysia, Singapore, Vietnam, Indonesia, and also in Philippines. So however, when we implement the pharmacogenetic testing, so we found that we screen the HLA-B1502, but the patient who negative of HLA-B1502, we prescribed carbamazepine for him, and he got Stevens-Johnson syndrome. So when we did the genotyping, and we found that this patient is 1521 carriers, but the HLA-B1521 is less frequency in Thai population, less than 1%. So therefore, we did not include that allele frequency, alleles genotyping into our national policy screening. Therefore, he got Stevens-Johnson syndrome. So therefore, in the clinical implementations, the HLA-B serotype 75 should be considered for the screening, not just only 1502. Moreover, when you look into the frequency of the HLA-B1502, you may see that in American, Europeans, Korean, and Japanese, even there are Asians populations, but there are less frequency of HLA-B1502. But they have their own study and found that the HLA-A3101 shows strong association with carbamazepine in Caucasians and also Korean and Japanese. So therefore, in the clinical setting, we should consider to combine the test for HLA-B and HLA-A to prevent the global populations. So this is I summarized the characteristic of the HLA-B1502 and carbamazepine for the practitioner preclinical practices. So I don't want to go to the details according to the time limitations, but you can read through by your materials. And let's move to the next one, which is the HLA-B5801. This is for allopurinol, not just only for Thai population as well, but we also have the we also share the biomarkers with the Asian population, including with Malaysia, Singapore, Vietnam, and also in Indonesians. So they have some study in Javanese from Yogyakarta study found the association with HLA-B5801 and allopurinol induced scar. And so you may have a question that about the other populations, including with Caucasians. And so you may see that HLA-B5801 distribute throughout the world. So therefore, by the study of the Caucasians, Korean and Japanese, they also show association with HLA-B5801 and allopurinol induced scar in that populations. Therefore, the HLA-B5801 can call as a universal biomarkers. It can use throughout the globally populations. And moreover, by the American Rheumatology Guideline, so from our study, we found that not just only the genetic factor, but we also found that non-genetic factor might be the predisposition factor that drive the patient to be scarred. For example, elderly patients, because they have lower function of renal function, and also renal impairments cases, and also drug interactions, and also the high dose of allopurinol treatments. Therefore, in 2020, American College of Rheumatology guideline recommend to prescribe allopurinol less than 100 milligrams a day if you did not screen for HLA-B 5801. And they are recommend to do the screening in the renal impairment patients because they have the underlying of the factor itself. And so this is, I summarize you about the characteristic of the HLA-B 5801 and give you in the materials. And also, we also have the study in a group of anti-HIV drugs, including with abacavir, and we show the HLA-B 5701 that is a biomarker for prevent abacavir to induce drug hypersensitivity. And also, we found the HLA-B 1301 for dapsone and chlorothiamoxazole. Chlorothiamoxazole is a common culprit drug for scar throughout the world. And so when you look into the structure of sulfamethoxazole and also dapsone, you may see that they are very similar structure. This is no doubt that why they share the biomarker between dapsone and also chlorothiamoxazole. In Indonesia, they have some study that recruit the patient from Papua, that is show the same biomarkers as well. Yeah, and so in Thailand, so we now implement as a national policy screening for HLA-B for 3 biomarkers, including with HLA-B 1502, HLA-B 5701, and HLA-B 5801 in Thailand. So just only one country that the patient no need to pay at all because all of the screening support by the government. Okay, so let's come back to answer these questions. So when we test the pharmacogenetics for their family, you may see that. So this guy who taking allopurinos, but they did not present the event like his mom because he did not carry HLA-B 5801 because he received this allele from his mom and this allele from his fathers. And we did the genotyping for our family here. And so therefore in this generation, they can take allopurinol without severe cutaneous adverse drug reactions. Okay. Let's move to the next approach of the pharmacogenetic because I have the limited times. Okay. So the next approach is the pharmacogenetics to adjust the dose or dose optimizations. So we can optimize the dose according to the metabolism rate of the patients. In the patient who have a genetic polymorphism on the metabolizing enzymes such as cytochrome P450, we can identify them as a poor metabolizer who is a low function of metabolism rate. If they are metabolism rate, they have very slow excretions. So therefore, the concentration of drug might be higher than toxicity levels. This is cause of the toxicity in the patients, and we can decrease dose to them to let them get drug within the therapeutic index. On the other hand, in the patient who are very rapid metabolism, they excrete very well. So if we prescribe as a standard dose guideline, so they may get like lower drug levels, lower efficacy level, which is cause of the treatment failure. We can increase dose for them. So this is the scenario that can be applied into the general medicine that you can see in the departments. So however, for some drugs, such as the clopidogrel, they have a scenario that different that I talked to you, but I will give you an example for a clopidogrel. In this slide, I summarize you about the pharmacogenetic study for dose optimization in Thai populations. And right now in Thailand, CYP2C19 for clopidogrel has been implemented as a national policy screening before prescribe clopidogrel in the patients, and also CYP2C9 for warfarin, and also TPMT and NUDT15 for azathioprine and 6-MP, which is a immunosuppressive agents. So I give you an example of the clopidogrel. Clopidogrel is an antiplatelet aggregation that we prescribe in the patient who got a stent to prevent stent thrombosis. So therefore, however, the clopidogrel itself is an inactive form. There are prodrugs. They need CYP2C19 chain to be active metabolite. So in the patient who have low function of CYP2C19, they may have low active metabolite and less efficacy. On the other hand, in the patient who have a very fast function of CYP2C19 or increased function of CYP2C19, they can change inactive metabolite to be active metabolite very well. So this patient may get efficacy very well. So therefore, in the patient who are low function of CYP2C19, clopidogrel is not the good choice for them. So in Thai populations, you may see that in Thai population we found 10%, about 7% that are poor metabolizer, and also 42% which is lower function of CYP2C19. So therefore 50% of Thai populations they may have less efficacy of clopidogrel if we prescribe clopidogrel as an alternative choice. So this is our case of the clopidogrel and the pharmacogenetics. So we did the stent for him, and so this patient got stent thrombosis, and so you may see that she got acute coronary stent thrombosis. She need to go back for surgery again within 1 week because we prescribe clopidogrel to prevent thrombosis, but it's not work very well in these patients. So therefore, we did the CYP2C19 for her, and we found that these patients is CYP2C19 carriers star 2 and star 3. Star 2 is no functions. Star 3 is also no functions. So therefore, this patient is a poor metabolizer. This is the answer that why this patient could not get efficacy well from clopidogrel, because CYP2C19 have no function, and it could not change clopidogrel from inactive metabolite to be active metabolite very well. So therefore, CYP2C19 has been implemented as a national policy screening in our populations right now. And so not just only for the genetics that should be considered. So as we test the gene, the pharmacogenetic gene is a germline gene that once in a lifetime, they have no change for the whole life of the patient, because that is a germline. You got that one from sperm and ovary from your father and your mothers. So however, when you look into these patients, so this patient, we prescribe Plavix, which is a clopidogrel brand name, and also aspirin as a Plavix in terms of the standard guideline. So after 6 months of the treatment, the patient come back to the hospital with bruise, which is a breeding sign of the clopidogrel or antiplatelet aggregation. So when you look into the gene, you found that CYP2C19 is a normal metabolizer. 2C9, which is a metabolism of aspirin, is a normal metabolizer. They should not have efficacy. They should not have toxicity in terms of the drug metabolism. So however, when we investigate in these patients, we found that after 6 months, she take supplements, which is from moringa powder. And by the moringa powder, moringa can induce CYP2C19 function and block the metabolism of aspirin. This is the answer that why this patient got breathing sign from Plavix treatments. So therefore, not just only the genetic, because genetic is once in a lifetime, no change at all, but lifestyle of the patient is a dynamics. So therefore, we have to follow up the patient for the whole life, not just only once in a visit. And so last of my case, so I would like to give you and emphasize you that in the clinical implementations, not just only for the pharmacogenetic testing, but the system that should be incorporated into the electronic medical record and also clinical implementations. Because this case, we already test for HLA-B1502 and found that she is a carrier of HLA-B1502 during she was admitted in the IPD wards. And so we avoid to prescribe carbamazepine for her until she discovered from that epilepsy conditions. And we discharge her from our hospital and let her come back to follow up at the OPD wards. And so when she come back to the OPD wards, she come to the another day that we make appointments. So therefore, she did not see the doctor that genotype for her and take care of her during the IPD. So the physicians, another physician looked at, okay, she could not control epilepsy very well. So therefore, they prescribed carbamazepine for her without no recognition about the genotyping in the health professional practitioner in this hospital. So therefore, I would like to emphasize you that in the clinical implementations, the electronic medical record should be set up and should be alert when the patient has been genotyped. And if they are positive of the biomarkers, the system should block for prescription such a drug in that patient. And also, the genetic counseling should be implemented for the implementation, because we believe that if the patients know their own risk, they can prevent themselves from the severe cutaneous adverse drug reactions. And during we do the during we did the genetic counseling, we give this opportunity to deliver the pharmacogenetic card for our patients. So you may see, this is a card that we should be delivered to the patient to let them recognize themselves from the risk of the drugs. And also, this is an example of the electronic medical record. When the patients, when the physician prescribes drugs, the alert system should be alert to request the genetic testing in that patient. And in case of the patient who are positive, the electronic medical record should block to prescribe such drug in that patient. And also, we can do the genetic counseling by the on-site and online. During the COVID-19, the patient don't want to come to the hospital. We can set up the online system as a telepharmacy to counseling the pharmacogenetic result to their patients. This is what we done in our country. And also, you may see that the pharmacogenetic information of the patient can be incorporated into the smartphone. And the patient can bring the smartphone to when they go to see the doctor or when they go to see the pharmacist in the pharmacy shop. And so in Thailand right now, the pharmacogenetic has been established very well in our country. But now we are moved forward to the next step to implement the pharmacogenetic in the community pharmacy. Because we know that if the patient get easier to get the pharmacogenetic testing, they can prevent themselves. So this is an example of the pilot study that we implement the pharmacogenetic testing in the community pharmacy in our country. And we hope that we can expand our system throughout the country. And also, last but not least, as you know that we test the germline, so and by the technology that whole genome sequencing or whole exome sequencing, once in a lifetime of the patients. So now the price of the whole exome is very cheap. And so we can do the whole exome sequencing and incorporate the genetic factor of the patient into the smartphone. And they can keep their own genetic risk, genetic information for the whole life of the patients. This is what we are doing in our country. And this is when we did the HLA genotyping, as Dr. Piroon mentioned. So we did the HLA-B genotyping, and we can see the primary finding and also secondary finding in the patients and let them avoid, for example, in this patient, they should avoid not just only for carbamazepine, but also allopurinol should be avoided in these patients. So you see, this is the power of the pharmacogenetics. And last of my slide, I would like to show you the impact of the pharmacogenetic screening in our country. You see that the number of the pharmacogenetic testing ordering by the physician is increasing year by year. On the other hand, the number of the Stevens-Johnson syndrome has been decreased for every year. We hope that we can eradicate and prevent our patient from the adverse effect such as Stevens-Johnson syndrome and toxic epidermal necrolysis in our Thai populations. And this is also my key message for you. And I would like to let you read through that conclusions. And I would like to emphasize you about the pharmacogenetics. It's not role of the not just role of the patient. Not just the role of the doctor or pharmacist, but it's interdisciplinary approach. So therefore, even you are not physician, you are not a pharmacist, you should be able to play your role in the pharmacogenetic clinical implementation and also precision medicine approach in the clinical setting. And so I would like to thank you very much for your attention. Yeah, thank you very much. I'm sorry that I'm talking very fast now because the times.
Okay, thank you very much for Associate Professor Chonlaphat. Thank you so much for mention about the interdisciplinary collaborations for or to increase the clinical outcomes of the patients. So we are already in the last session for the last speakers. We have Professor Chaur-Jong from Taipei Medical University. Yes. Okay. Hello, yes. Professor.
Hello. Yeah.
Okay. Hello.
Good afternoon.
So, yeah. Good afternoon. And you have 30 minutes for your talk about the biomarkers, right? And pharmacogenomics aspects in neurovascular disease. Is it right? Okay.
Yeah.
So, yeah. So I'll give the time to you. It's about 30 minutes, please.
Thank you so much. Okay. Thank you so much for your introduction, and it's my great honor to present my talk here. Actually, I have visited UGM a few years ago, and I stayed there for 2 days. It's a beautiful university. Today, I want to talk about the precision medicine in neurovascular disease. I will focus on biomarkers and pharmacogenetics. First, I would like to share our study about precision medicine in dementia. As we know, dementia actually is symptoms instead of a disease. Many diseases can cause dementia. If the patients present a cognitive decline and the cognitive problem affects the daily activity, we can diagnose the patient with dementia. But the underlying disease can be many kinds of disease. The most common one is Alzheimer's disease. I believe you have learned this disease very much. And the second one, especially in Asian country, is the vascular dementia. In terms of the dementias caused by stroke or cerebrovascular disease. And the third one is dementia with Lewy bodies. The patients can present dementia in addition to the movement disorder like Parkinsonism. And the fourth one is frontotemporal dementia. The patient would not present memory decline in the initial state, but show the personality change, mood change, disinhibition, or some kinds of language difficult. So there are so many kinds of neurodegenerative disease, but most of them are caused by abnormal protein accumulation in the brain, such as amyloidopathy. In terms of the amyloid beta accumulate in the brain, it's Alzheimer's disease. And there could be alpha-synucleinopathy, like Parkinson's disease, dementia with Lewy body, multiple system atrophy. And also we can find some patients are tauopathy, like frontotemporal dementia, cortical-basal ganglion degeneration, or progressive supranucleopathy. Actually, beyond these three common forms of abnormal protein accumulation, we also can see some patients are induced by TPP43, like motor neuron disease. So we can say that most of the neurodegenerative disease are caused by abnormal protein accumulation in the nerve system. So we need to dissect or diagnose the disease precisely. In the clinical, we can have a clinical history taking or clinical evaluation. We also need the neurocognitive test to define the cognitive decline. And we have a lot of neuroimaging, like MRI, PET scan, or SPECT scan. Right now, we have a so-called molecular image. We can measure the abnormal protein, like amyloid beta accumulation in the brain before the patient death. So right now, we diagnose the dementia patients depends on a lot of clinical tools. I think the molecular image, like amyloid PET, is very powerful because since last year and this year, there are two monoclonal antibody drugs can remove the amyloid beta from the brain for the Alzheimer's disease patients. So in the future, I believe molecular imaging will become more and more important for the precision medicine. And as we know, there are a lot of wearable devices. So a lot of digital biomarkers has been developing. Like we can measure the patient's sleep, gait, or movement, and even the speech, tongue, and the speed. So right now, we also conduct a few studies focused on the digital biomarkers to help us to predict or diagnose the patient disease precisely. I think the previous speakers has talked about the genetic and the genomics, and for the nerve system disease, we also can make the survey for cerebrospinal fluid, CSF, because the CSF is quite near the brain. So we can get the CSF and to do the survey, and we can find some biomarkers can help us to diagnose the disease. Currently, the very hot topic is so-called blood-based biomarker for the neurodegenerative disease. I will talk this issue later. The other hot topic is about the gut microbiota, because a lot of reports have shown gut microbiota in terms of bacteria content in the stool can help us to differentiate the disease. And we also conduct a study about oral microbiota. It can show the very similar results to the gut microbiota. This figure shows the natural cause of Alzheimer's disease. You can see the first step, even in the cognitive normal state, the amyloid beta has accumulated in the brain. Follow the A beta accumulation, the tau protein appear in the brain, and then we can see the brain atrophy. And following the clinical presentation, the first initial presentation are memory decline, and then the cognitive decline affects the daily activity. We can say this stage, the patient with dementia. So this is the biomarker we call the ATN system for the diagnosis of Alzheimer's disease. A is amyloid. We can check the CSF amyloid concentration. We can use the amyloid PET. For tau, we can use the CSF phosphorylated tau concentration, and we can use the tau PET. For the neurodegenerative presentation, we can use the CSF total tau and the neurofilament light chain. We can use the metabolic PET, like FDG PET, and also we can see the brain atrophy by MRI. So we can see the current biomarker of Alzheimer's disease include MRI, FDG PET, amyloid PET, and the CSF study. But these biomarkers are not very useful because everyone has a disadvantage. Like MRI, it only shows in the late stage, and FDG PET is non-specific, and amyloid PET is very expensive, and the CSF study is quite invasive. Most of our patients refuse to undergo the CSF study. So a blood-based biomarker is necessary. But there are some difficulties for blood-based biomarker. First, as we know, the brain and between the brain and the blood, there are so-called blood-brain barrier. It's a tight junction and separate the brain and blood. So the biomarker from the brain, it's not easy to get into the blood. And some studies showed the blood biomarker has a dynamic, diurnal change. When the patients are awake and asleep, the concentrations are not the same. And as we know, the CSF can separate the brain and the blood. So some biomarker have to move by active transport or diffusion. And I think the most important is a lot of biomarkers concentration are high in the CSF, but very, very low in the blood. So there are a lot of tools has been tried to measure the blood-based biomarker, including immunoassay, molecular assay, and they try to make a panel but not very successful. This is our study. We use the so-called magnetic reduction immunity. We coding the antibody, amyloid antibody, on the bead with the magnetic, and we let the beads into the magnetic field, and we can make a so-called ultra-sensitive method to measure the concentration of amyloid and tau. And then this is a clinical trial. We can find the CSF, the early stage AD has the highest concentration of amyloid and tau, comparing with the cognitive normal population individuals. We also can conduct the genotyping for the patients. As we know, the ApoE4 area is a risk factor for AD. This is a study published in Sains. You can see if the patient has two areas of ApoE4, the AD will occur earlier and younger and more severe. So it's like a dose-dependent manner. This is our study. We also found the ApoE4 carrier have a 5 times high risk. So there are 3 genes mutation can cause AD, but ApoE4 is a genetic risk factor for AD. Right now, a lot of research group developed the so-called polygenic risk score. They measured a panel of many genes and calculate the genetic risk. This is a very good, example to show if the polygenic risk is low, the patient will have a low risk to develop the Alzheimer's disease. If this line is ApoE4 carrier, and this line is to represent the high genetic risk. This study shows if we subgroup according to the genetic risk, in the high genetic risk group, if the individual have a good lifestyle, the risk can be reduced. I think this is a very important point. If the patient contains a high risk genetic, high genetic risk, the people should have a good lifestyle. This is our another study. We study the gut microbiota. We collect the stool from the patients, and including dementia, mild cognitive impairment, and the normal. You can see in the stool from dementia, they have a high percentage of firmicutes and the low percentage of bacteroides. So you can see among these 3 groups, the F-B ratios are 5 and 1. We also conduct the oral microbiota study. We got a very similar results. The firmicutes bacteroid ratio are high in the AD group. And then I will move to the pharmacogenetics or pharmacogenomics. I believe this part has been discussed very comprehensively. So I will go this part very quickly. This is a national side effect report system in Taiwan. You can see there are about 200 patients with a side effect of drug has been reported. The most common one is skin and subcutaneous tissue disorder like Steven Johnson and toxic epidermoid necrosis. So you can see the most important or most common one is skin damage. There are also nervous system disorder like neuroleptic macular syndrome. Among these patients, the allopurinol is the number one. The second one is phenytoin and anti-TB drug. And also, this is, you may heard a lot, carbamazepine, and also some painkiller like chlorpheniramine. So, talking about the first effect of drug, it could be dose dependent, it's related to metabolism, and the second one is dose independent. As we know, the drug metabolism will involve, the first step is oxidation reaction by the cytochrome P450 system. Cytochrome P450 it's include about 57 genes. There are few important enzymes of cytochrome P450, including the CYP2C9, 2C19, and the 2D6. They are responsible to the specific drug metabolism. This is an example. Warfarin is anticoagulant. So the most dangerous side effect is bleeding if we give the too much or too high dose of warfarin to the patients. Yeah, actually the warfarin was digested by the CYP2C9, and you has now start 2 area and the start 3 area are poor metabolizer, and you can see the patient is the patient with normal metabolizer in the community. They are around 0.6, but for the warfarin low dose, they are around 0.19. So it indicates the patient with poor metabolizer, they will shift to the low dose. The other example is coperigot metabolism. As we know, coperigot is for drug. It needs to be digested by 2C19 to become an active form. So in Taiwan, there are about 51 to 57% population has poor metabolizer. Yeah, this is our study. We compare the coperigot and aspirin. These 2 drugs are both a standard drug for secondary prevention of ischemic stroke. you can see the actually among the patients in Taiwan, the clopidogrel is not good compared to the aspirin. This may be due to there are about 50% Taiwanese people belonging to the poor metabolizer. The other one is dose independent. I think is come to due to the immune reaction, and HLA typing should play an important role. This is a clinical trial conducted in Taiwan. We joined this trial, and the study design is if the patient wants to take carbamazepine, they should take the genetic test first, and we can, the research team will tell the physician the genotype within 3 days. So the physician can decide give the patient carbamazepine or not. You can see there are about 372 patients are HLA-B1502 positive. The other one, about 4,000 are negative. Demographic data are the same. However, among this is history data. You can see in Taiwan, there are about 100 patients with Steven Johnson or skin reaction before this trial. Following the study design, there are no patients suffer from the Steven Johnson or 10 in this study. So this is this study published in New England Journal of Medicine. It's very important result. So in Taiwan, Taiwan Health Insurance request HLA-B1502 tested before use the carbamazepine. So I would like to close my talk here. The take-home message is the precision medicine integration of multimodal biomarker provides pharmacophysiology-based diagnosis and the treatment. Pharmacogenomics or pharmacogenetic help prevention of diverse effects of drug, both for dose-dependent and dose-independent mechanism. Thank you.
Thank you so much for Professor Chaur-Jong. So based on your presentation, yeah, it's important to know the biomarkers and the pharmacogenomics aspects to improve the clinical outcomes of the patient who has the neurovascular disease. So all of the speakers already have finished their presentation, and we have about 30 minutes for the questions and answer session. Is there any question from the participant here? Okay, we already have one. And the participant who joined the summer course by online, you can write down your answer in the chat box. Okay, so we already. Okay, yeah. Please go to the mic. Okay, you can state your name and then your questions.
My name is Zulfa. I'm from Medicine UGM 2019. I would like to ask a question for Dr. Ruzilawati. Can I ask more than one question? Oh, yes.
Of course.
For the first question, considering that there are a stigma surrounding drug addiction or mental illness in general, if there is a genetic predisposition, how do you fight negative stigma in community to reduce racial and distinct socioeconomic biases, considering that there are complex environmental and societal influence factors, and we do not want to, I do not want to say reductive, and I do not want to put all for one fit, all or all people fit in one box. So I would like to know, how do you fight the negative stigma in community, considering that I have mentioned. And for the second question is that since considering the same thing and the findings that are possibly can only be applied to the study population, what can we expect and how do we improve research in the future to avoid again the racial biases in etc., since Indonesia is consisted of diverse ethnicity and racial and races. And then the third one is, how do you expect governmental rehab facilities to support more precision healthcare? Thank you.
Sorry, I'm not clear with the first question.
Oh, the first question, okay. Considering that it's not to say that there is always a genetic predisposition in addiction or smoking behavior, but if there is a genetic predisposition, how do we fight the negative stigma in the community surrounding drug addiction to reduce racial and distinct socioeconomic biases?
Okay. Usually for addiction, there are other factors that influence the smoker to smoke, not only genetics. Usually, meaning that there are other factors that should be considered in smoking behavior. For
the second question is what can we expect and how do we improve research? I would like a practical answer in the future, since the findings possibly can only be applied to the study population.
I mentioned in my lecture that different ethnic, they give different genotyping patterns. So we need to study those genotypes in those population that we want to study.
So we take the sample population.
Yes.
In refer to this population. For example, if you want to refer the sample of Indonesia, so we take the sample for Indonesia.
Yes.
Okay. And then the last one. What are the practical example of improvement or development, or how do you expect government rehab facilities to support more precision healthcare? For example, for patients with drug addiction, they more resort to prison rather than rehab facilities here, unfortunately. So can we apply precision healthcare also in governmental facility?
So for precision medicine in addiction, they need usually we need a long way to go, because we don't have any evidence base to confirm that precision medicine can be used in addiction treatment, not like HLA. So we need further research need to be performed.
Thank you for the presentation and for the question, Doctor.
Okay,
So
is it very enough,
add more
Zulfa?
information for you? So as a national policy screening or a national implementations, so as even like we did the HLA genotyping, but that is not very, it could not be jumped from the first study to be national policy screening. The important thing that cost effectiveness analysis in the country, you have to prove that if you do the screening as a national policy screening, it's cost effective to save your budget of the public health budget, not jump from the first study. Even like for HLA-B1502, we did 10 years from the first study to be the national policy screening, we did the retrospective study, several study in independent cohort. And then we moved to prospective study to prove that two arms, screening and non-screening, and it can prevent the Stevens-Johnson syndrome from the screening arms. And from that, we developed the guideline, local guideline and also international guideline, because you have to prepare that, because if you implement as a national policy screening, the first day that you implement, you need to have like guideline for physicians and pharmacists, for example. So, and then we prove as a cost effectiveness, and we found that it's positive. So therefore, all informations package, incorporate as a package and submit to the policy maker, and then they decide to implement as a national policy screening. I would say that it's very long time for translational from research to the clinical implementations. Yeah.
Okay. Yeah. So we need the long period to implement the precision medicine, right? Okay.
And also in the clinical implementations. So you have to prepare the education of the health professionals, because as you may see that in my case, the last case, the doctor who did not recognize about the HLA-B 1502 in that patient, even she got the screening, but the doctors did the prescription for her. That is the knowledge limitations for the health professionals that should be provided, as a national course on a national curriculum. Yeah.
Yes. Okay. Yeah. Maybe based on my experience, because I'm a pharmacist, we just only know the theory that this drug is induced the metabolism of other drugs, but without any pharmacogenetics screening, we don't know which the specific gene that patients have at that time. Okay, so is there any question from the other participant, or okay, please.
Okay, my name is Talia. I would like to ask Professor Sukasem, actually relating to what you just explained about how long it takes from the first study to then becoming a national policy. I wanted to ask for patients and populations who are in areas that are harder to reach. For example, in Indonesia, because we are an archipelago and there's such, we have such a spread out population. In trying to actualize a national policy screening like that, it would be very hard to reach areas, say, from Java to Papua. How was it like in the 10 years that it took for that national policy to happen? Like, what was the process and what was the difficulty, especially in reaching patients in harder to reach areas? Thank you.
Thank you so much. So as I already mentioned, that it's so hard to translate from the research into the clinical national policies implementations. So for example, HLA-B 1502, as you talk about the diversity populations in Indonesia, right? So in our in our study, so we recruit the sample from throughout the country, systemically recruitment, and represent to each populations, and we prove that in Thai populations we are homogeneity. The frequency of the HLA-B 1502 is about 16% throughout the country. But also for HLA-B 1521, that is a biomarker for guys who got Stevens-Johnson syndrome. That is a serotype 75. Actually, they are the same structure of the proteins, but different alleles. So the HLA-B 1521, that is less than 1% in our populations, but it's high frequency in the southern part. That inference from the population from Malayu or Malaya, something like that, in Malaysian populations. That is the influence of the populations. HLA-B 1521 is higher than 1% in that southern part of our country. However, if at the first phase that we implement, we focus on the majority of the screening, 1502, that is majority in our populations, and we could not cover, the test could not cover for all genotyping, including with 1521, because if we would like to screen for all biomarkers for carbamazepine, the cost of the test might be higher and the cost effectiveness might be negative. So therefore we have to let them go by by their own destination, something like that. But however, right now, fortunately that we have the new technology, like Dr. Piroon said, and we also have the new test that can cover for all alleles, including with serotype 75 for all alleles with a very cheap test, a very cheap price. The test is about actually $10 to cover for all alleles. This is right now we can cover for all, even there are less frequency in our populations.
Okay. So yeah, is it clear enough, or maybe you wanna ask more?
Yeah, I'm sorry, just like a little follow-up question. From the first time that the genotyping became like a national screening test, how long did it take for it to be implemented like across the country? That's all.
So you mean at the first time that we implement as a national policy screening, right? And how long that we take throughout the country, right?
Yes.
At the first day.
Wow.
Because we have to prepare, get ready before we implement as a national policy screening. We have to prepare the technology, transfer our technology throughout the country to prepare the laboratory. And we have to prepare the health professional knowledge, educations as well.
Oh, so it was like launched nationwide on the first day?
Yes, yes.
Thank you so much.
Yeah.
Okay. Yep.
Okay.
I would say that you could not discriminate the people, even they are the same, under the same roles, under the same insurance, national insurance. Yeah.
Okay. So, okay, thank you for your answer. And then any other questions? Okay, please.
Good afternoon. I want to ask regarding the national screening in Thai, especially the HLA-B 1502 and the 5801, right? So what method is being used? Is it PCR or sequencing? And which method do you think is much more better to be used for national screening? Okay, in terms of accessibility and financially. Okay, thank you.
Or yeah, maybe
Okay.
you can
Sure.
answer
Okay.
first.
As I mentioned that the cost of the test is a factor for a national policy implementation. So therefore we try to do cheap as much as possible. So at the first time we provide a service and about $30 baht for one test. And that is acceptable for a national implementations. And limitation of that test, as I mentioned, we lost of some of the patients who carry serotype 75. So until right now, we improve the technology and include that genotype, include that alleles into our test detections. So therefore, the test is cheaper and is better for the detections. That is what we are doing. So as you talk about how hard to do the genotyping, right? So we can do the very basic technology like PCR, real-time PCR. That is very easy and very rapid because we know that the physician waiting for the result before prescriptions. So therefore, we try to return the the result within 1 day, 24 hours. So actually, the test can be finished within 3 hours, actually. But we have another process for reporting and approve the result by the pharmacists and also medical technologists before launch the result to turn to the physicians. That might take another time. So therefore, we guarantee to test to return the result within 24 hours. Yeah,
Maybe
it's very easy to detection, I think.
okay. Yes. Maybe Dr. Piroon, you have.
Yeah, so in terms of the technology that I present today is compared with the technique that in that implement right now is more expensive, definitely, because of its the sequencing and it's the new technology. So for the research right now is good because you can go very detail of the and resolution is very high. Yeah, but in terms of implementation, it's not there yet. So you need to have like more sample to do barcode together and sequence at the same time to lower the cost, for example.
Okay. Yes. Thank you. Is it clear? Okay. Okay. Yeah. We have another questions. Please.
Hello, I'm Raihan. So just now, Prof and Doctor have explained that how people respond differently to the drug that can cause adverse side effects. So I'm interested to know whether the allergy also works in the same mechanism that involve HLA.
Which drug that you mentioned?
Ah,
So
sorry.
I'm sorry. What about your question? I'm sorry.
Oh, I want to know whether the allergy works as the same mechanism as HLA.
Yeah, that should be because the allergy that is from allergen or antigens, that should be the same process. But for some allergen, we don't know what is the specific HLA yet. So therefore, right now, we could not screen the HLA genotyping for the allergen. Yeah, but that should be the same mechanism because the HLA is the protein that present antigen to the T cell receptor to activate the immune system.
Can I add?
Anyway, so you have to keep in mind that allergy, they have many type of allergy. Like in my talk, we talk about delayed thyroid hypersensitivity. But if you talk about angioedema, that is like edema of like nose or your throat, that might be a different mechanism.
Okay. Yeah. So I have another question related to that. Does the HLA expression decline with age, like asthma do when, for example, if a person have asthma since young, and then when they become older, they will adapt to it. So does the HLA has the similar, like, decline in the expression? Yeah, thank you. Thank you.
So thank you so much. So because as I mentioned that the HLA is a germline, there has no change at all. And so actually, even you are grow up, the HLA is to be expressed on every cell that have nucleus. So sometimes if you have like normal situations, you have no antigens, HLA have their own functions. They should express and present the cell peptide to prevent your cell as a negative selections. That's why they did not decrease expressions as well as actually. I don't know that I answer your question. Yeah.
Okay, maybe from the left side here, is there any questions from the students? No? Really? Okay, so how about from the online participants? Is there any questions? Okay, while we are waiting for the other questions, maybe I'd like to go to Prof. Chaur-Jong. Yeah, I'd like to ask you about the implementation of the pharmacogenomic screening at Taipei. Could you share or could you tell to us about the implementation there?
Yeah, there is a national health insurance system in Taiwan. So over 99% Taiwanese are covered by the national health insurance. So the national health insurance requests the HLA B sorry, 1302 genotyping before the prescription of carbamazepine. So I think right now there is no Steven Johnson syndrome patients due to the carbamazepine use. Actually, since next year, carbamazepine will withdraw from our Taiwanese market because the genotyping there is a little trouble. Less and less use of carbamazepine in Taiwan. So the drug company decided to withdraw this drug from Taiwan market. Yeah.
Yeah. Okay. Thank you very much for your sharing, Prof. Okay. From the committee, we already have the questions from the answer from the online participant. Okay. From Claudia. Okay. Hi, Claudia. So. Thank you to all the speakers for the very interesting talk on pharmacogenomics and precision medicine. So the questions is about, this question is for Prof. Chonlaphat. Could you share any challenges in genetic counseling for pharmacogenomics in your setting? Yeah.
Thank you so much. So actually, in every case, by our regulations in our country, so in every genetic testing, we need to do pre-counseling and post-counseling. By the pre-counseling, we get the consent form of the patient for every patient. And by the post-counseling, we have to do, as you may see, like in that case that we need to do post-counseling in the patient who got toxic epidermal necrolysis. So usually the question from the family that why you don't screen before, why you screen after, that is also the challenge of us. And also another thing is that I think the genetic counselor is so limited in every country. So therefore, the genetic counselor course has been established in our country. And at the first phase, we developed the course for a genetic, to develop the genetic counselor, to build up the genetic counselor, which we developed as a short course training, 4 months. So actually, we should expand from 4 months to be like master with 2 years, for example. That can be the full genetic counselor. But at the first time, we have no genetic counselor in our country. So therefore, we have to rush to develop the genetic counselor in our country. And another thing that is very joking for me, because as you may see the genetic HLA genotyping of the family, if you can remember, you can use that test as a paternity test, you see. But actually by the pharmacogenomics, we should not mention out of the scope of the pharmacogenomics, because sometimes you may did family genotyping and you found that they are not real paternity, but you don't know about the background of that family yet, right? So you should not mention about that out of your scope. You just mention that about the pharmacogenomics. And also, in the pharmacogenetic testing, we can use that testing to predict some diseases, such as ankylosing spondylitis, which is a biomarker of ankylosing spondylitis, is HLA-B27. That is the first field of Dr. Piroon mentioned. So you can see in the patient who test for allopurinol or carbamazepine, we can predict diseases in that patient, but it's out of our scope. If you mention about the disease, risk of the disease in that patient, next year this patient have to pay more money for their own insurance, because they have their own risk by the genetics. You get my point? So therefore, this is not the scope that we have to mention when we do the genetic counseling in the patients. Yeah, that is also the challenging.
Okay, that's all the answer from Prof. Chonlaphat. Claudia, maybe, is it clear enough for the answer? Any comment from you? Claudia, are you still there?
It's very clear. Thank you, Prof.
Pardon?
Yeah, no, it's very clear. Thank you, Prof.
Yeah, okay. Thank you for your questions. And then maybe the other questions from the online participant. Is there any questions? Okay, how about the other participant here, maybe from the left side here, from the student from Universiti, right? Okay, is there any questions? No? Okay.
I have some more information. So about the genetic counseling course in our country, we now develop as an international course that we can accept for the international students. Yeah.
Oh, okay, that's interesting. Okay, thank you for your information here. I'm curious about how about in Malaysia? Is there any genetic counselor also, or? Okay. Yes.
Usually the genetic counselor play role in the genetic disease testing, not for pharmacogenomics. Yeah.
Okay.
I think that in every country you have like genetic counselor, but not too many people. But they pay role focusing on the genetic disease, especially for rare diseases.
Rare diseases.
Yeah.
Okay. Yep. Thank you. We still have. Oh. Yeah, we still have 3 minutes left, maybe, if there is still any questions from the participant here. Okay, so, yeah. To conclude this session, so all the speakers already shared their knowledge to us about the pharmacogenomics from the theoretic from the theory about the pharmacogenomics itself, and then how to process the pharmacogenetics test, yeah, and then the implementation, and also the importance of knowing the biomarkers and pharmacogenomics aspects in the neurovascular diseases. So thank you so much for all the speakers. Thank you, Prof. Chaur-Jong. Okay, please give applause to all the speakers. Okay, I do hope you can get new knowledge and a lot of insight from this session. Thank you so much for your attention, and yeah, good afternoon, everyone. Thank you so much for joining this session today. Yeah, thank you so much.
Okay, so thank you so much, Butia. Can we take a photo together
Okay,
for all the
please.
members of
Yeah.
panel 1? Mas Itam, can you help me to take a photo? Okay, Prof. Chaur-Jong, can you turn on your camera for the photo sessions? Mas Dayan, can you show the Zoom display on the screen? Oke, gimana enaknya Mas? Maju, agak maju gimana? Agak tutupan. Oke, agak geser dikit ke tengah mungkin Ibu, biar kelihatan. Jadi geser ke kanan kiri gitu, biar yang tengah kelihatan. Oh ya, oke. Ah yes, that one. Okay, so yep, thank you so much. And once again, guys, give applause for our member person for panel 1. And that was the first day of summer course 2023. And tomorrow we'll start for the second and third panel. And tomorrow, make sure you are already in the auditorium at 8 a.m. And I also give you some information. You already have one task in your ELOC or learning management system, so make sure you check your ELOC for the assignment. Okay, so thank you so much for coming. Wassalamualaikum warahmatullahi wabarakatuh, and see you tomorrow, everyone.